Hi, everybody. Welcome to another Chats with Chaudhry. Today, I'm delighted to be joined by David Hipkiss, who is CEO of Enesi Pharma. And David presented a few weeks ago at the DDF Summit Europe, which seems like months ago now, but it was only a few weeks ago, on ImplaVax. And this is quite a long title, so bear with me. In vitro and in vivo data confirmation of thermal stability and immune responses using a wide range of injectable unit solid dose vaccines constructs. And today we're going to learn more about ImplaVax and the technology around it. So that's such a long title, David, but lovely to meet you. How are you?
Yeah, I'm great. And sorry for the long title there. It is actually quite long, isn't it? But it did capture an awful lot of things. I think we wanted at the show to really highlight the breadth and the depths of what we're doing and evidence that by data, of course. And once you've got that data, that starts to lead into a few other questions. So we were delighted to be part of the event and looking forward to following up talking with you today.
Fantastic. So before we start, whereabouts are you based then? Where are you calling in from today?
Just north of Oxford.
All right. So hopefully the sun is shining as it is here in Surrey today.
Well, the sun always shines in Oxford, at least underneath here, not about Surrey. But no, it's a beautiful day here today. We had a lot of rain yesterday, but it's great today.
Fantastic. Right. So before we start talking about ImplaVax and the technology around it, would you mind giving viewers a very quick overview of your background and also maybe a bit more detail about the business itself?
Yeah, thanks. I'm David, historically trained as a chemical engineer. Well, that seems a very long time ago. And I've been in the life science industry now for actually getting on for 30 years. Gosh, where's all that time gone? I've been with big companies and small companies. I've always been associated and led companies of sort of combination products, particle engineering and devices spanning respiratory therapy, chiral technology. Founded a few businesses, raised some money, sold a few as well. And in 2017, I was the CEO and founder of Enesi Pharma, which is making great strides in the worlds of vaccines and vaccination. So a little bit about what we do before we get onto the technology. Enesi is a unique company in the world. And we put vaccines into a novel form into solid state. So little micro implants, which are smaller than a grain of rice. And then we put these into subjects and into patients via way of means of a very simple, very cost-effective actuator. The technology is designed to solve many of the innate problems associated with needle and syringes. And we work with quite a number of people around the world, be they big pharma, specialty pharma, governments, NGOs, and actually a few of our own product developments as well. So to put us in context and sort of size, we're a little over 30, 35 people right now. We have laboratories based down in Milton Park in the south of Oxfordshire, and have come a very long way over the last 15 months. In fact, we've doubled in size during the COVID pandemic, which might give you a flavor for the type of things we're getting involved with now.
Right. Well, thank you for that overview. So you said that Enesi is the only company in the world pursuing this approach then?
Yeah, we are. I think one of the benefits of the pandemic, if there is one, I have to say, apart from collaboration and regulatory science coming together and a willingness to work in ways which have hitherto been unparalleled, is in the vaccine space is very much back in vogue. You know, historically it was a little unloved. People didn't really understand the value of vaccines, and you always did what you've done, so you always got what you've got, be that the way you made a bulk vaccine or the way that you delivered the vaccine. Right. But now with COVID, sort of the spotlight's been put on the challenges of logistics and deployment and administration. And I think that's enabled a little bit of a renaissance for companies like Enesi to take their novel delivery technology forward back onto a window on the world. So there are a number of ways of vaccine delivery being pursued over and above needle and syringes. You could think of maybe oral electroporation, microneedle patches, intranasal. But what we do is a little bit unique, and is that we're the only company in the world which is pursuing a needle-free but injectable unit solid dose presentation of a vaccine. And that's a little bit of an oxymoron, if you will, needle-free but injectable, but it is. And if you can imagine, we're essentially taking what would be a bolus of liquid vaccine, turning that into a precision-engineered solid monolith, and then giving that to the subject for a range of benefits.
Right. So I was going to ask you about what you need, but you've already covered that. So can we go in a bit more in depth of how it actually works?
Yeah, so that's a really good question. Maybe borrow some things around as well. So, being a chemical engineer, we don't like to reinvent things if we don't need to. And if things work quite well, that's quite a good thing to basis to build your technology platform on. So the first thing that we do is we take a bulk vaccine, which we're given. So we're not a vaccine discovery company, and we powderize that vaccine via standard methodology. And standard methodology for powderizing biologics are typically lyophilization with sugars, or spray drying with sugars. So the first part of our processing is one of those two things, depending on the vaccine construct. We then take that powderized vaccine and we impart and mix it with some other excipients, typically for thermal stabilization, but a lot for structural strength as well. And then we use a couple of processes, be that cold extrusion, and that really is like spaghetti making, to be honest. It really is. It's spaghetti making. Or a little bit of higher end micro tableting, with the goal to end up with unit doses of solid vaccine, where each and every dose contains the right amount of vaccine or vaccines, combinations or adjuvants thereof. So that's how we make stuff. Because we use the well-proven technologies of lyophilization and spray drying, thermal stability is sort of a given, really. But we do have to manage that process through each and every step of the production, right? Because, as you know, if you touch things in a vaccine world, they can have a negative effect on potency. So we have to be very careful about that. Sure. And also we have to do that in an aseptic fashion. So now we've got our solid vaccine. How does it work? So the first thing we do, we impart it into a really nice area of the skin. So we're not intradermal, neither are we intramuscular, but the solid doses are pushed into the subject about 3 millimeters below the surface of the skin, and they extend to about 7 millimeters below the surface of the skin. So we like to use the term sort of transcutaneous. It's a bit Hovis, really. It's a bit of best of both, best of all worlds. And that area of the skin is packed full of biology that really helps you in terms of antigen presenting cells and the mechanisms of actions that need to then go and transcribe whatever you've put there into making proteins or antigens or T cells, which is how the vaccines work.
Can I ask you a question about that just very
quickly?
Yeah, sure.
So
well, two questions as a layperson just listening to this is, where part in the body would you do the delivery, and secondly, what sort of mechanism do you use then to deliver it? Because you said it's not a needle, so how do you deliver that then into the skin below, into that depth?
Well, with our special actuator, I should have brought one. I might actually have one in my bag behind me if I'm lucky. If not, I will have to go upstairs and get one. No. All right. No. So what we do. So I think that the key is we don't introduce any change in macro clinical practice whatsoever, as opposed to needle and syringe. So we will go into the deltoid, we'll go into the thigh or the buttocks, depending on the subject, really.
Sure.
The implant itself, imagine it like a little micro depot, almost like a mini projectile, because it has a sharp tip on the front. It's held within a little cassette, which you click into the spring-powered actuator and lock and load, and then simply press down, and then the mechanics within the actuator fire a spring-powered plunger, which pushes the back of the implant and pushes it into the subject to the same depth each and every time, independent of administration site or subject. So hopefully it's an injection mechanism in exactly the same way as a needle and syringe would do, but we don't need the needle and syringe because the implant, if you will, is sharp and it replaces the needle in its own right. So if that makes sort of sense.
You know, visualizing Star Trek now, actually, to be honest.
well, Star Trek, that got to be careful there, Raz, actually, because Star Trek was, there were some people in history who tried to do the Star Trek one, and they used a gas-powered gun to do that.
Right.
But it's a very interesting analogy, and they sort of a little bit blazed the trail, but they had a powder bed which had potent material on heavy particles. It was gold particles. It was powder jet. And then they fired a gas under high pressure to entrain the powder through a nozzle to then get into the skin.
Right.
A couple of big changes and challenges in physics there, particularly about repeatability. And of course, once you've fired the gas once, where do you get the next lot of gas from?
Sure.
Our spring-powered actuator can be reused 1,000 times, and we don't have the variability associated with that type of technique. So very interesting precedent set many years ago, actually, for the benefits of a solid dose vaccine. But what we've done is probably more super gun than space gun. I think that's probably a good way to describe it.
Right. Okay. So you carry on. So I interrupted you.
Yeah, no. So it's a really good question, because we're targeting the right areas of the skin. So then listeners need to think about that little micro projectile. It is like a solid monolith, really, but it's a solid monolith of microparticulate material. And microparticulate vaccines are known to be highly immunogenic. But then if you think a little bit more engineering-wise, you've put a dry substance into an aqueous system. And of course, through reverse osmosis, well, you've got to draw liquid towards it. The liquid contains all of the antigen-presenting cells you want. It's going, what the hell's this? Don't know what this is. Okay. It has to attack it and eat it. And of course, it swells a little bit. It takes up some moisture. That deals with a little bit of local reactogenicity, not too much, not too little. So it's quite Goldilocks in that regard. But then what we find is, because our implants act as like a depot, we can also control the rate of how the vaccine releases to the body. So rather than being drained quickly away through the lymph system as if it was an intramuscular injection, our vaccine implants tend to hang around a little bit. So we have the technology to make a bolus release, so they'll dissolve in a matter of minutes. We can delay the release, so maybe they won't come on for a few hours or a few days. We can sustain the release over an extended period of time. And some of our more recent work is really at the edge of science, where we have a pulsatile release system. So we do a burst release, some immediately, and a burst release sometimes later. And that then starts to get into quite important topics for making vaccines work very well, which is about adherence to regimen. And actually, quite frankly, as COVID's shown us, it's not the first dose that necessarily is the challenge, provided you can get the logistics. It's getting people to come back on time each time for the second one. And for a 3-dose regimen, that's really difficult. For a 4-dose regimen, it's really impossible, unless you're a little tiddly one when your mum's taking you there when you're young. So the ability to almost do a prime and boost either in a single shot or in a single visit with different types of technology, I think there's an awful lot of opportunity there to make a real difference on healthcare worldwide.
Well, it sounds amazing. I mean, one question which comes to mind when you're talking about the vaccines, is there a difference though, or does the type of vaccine impact how effective it is? Because obviously you've got just with COVID, you've got the AstraZeneca ones and the Johnson & Johnson ones, then you've got the mRNA ones, which obviously Moderna and Pfizer. And so depending on the type of vaccine, how it's been created, does that impact the delivery mechanism and the efficacy or the effectiveness of the method?
Okay, so if we had 5 hours, we could talk about that. I'll give you the vox pop on it. And I think there are 2 answers to it. And one is about logistics and one is about manufacturability and maybe how things work. So let's take a step back. Look, hats off to Pfizer, Moderna, BioNTech, AstraZeneca. Amazing job that they've done. And actually what you've seen with regards to the prime difference between the mRNA and the viral vector approaches, AstraZeneca, Oxford, J&J, and the others, is essentially how they're delivering a message to the body to transcribe to make antibodies and T cells. So the viral vector is essentially a carrier. And historically, other carriers were used. There may have been whole cells, there may have been subunits and fractions, and they had to get intracellular, but it was a carrier. And I think when we first saw the early results, both from Pfizer and Astra, it essentially gave a proof of mechanism that the attacking the spike protein was a viable method of doing it. So that was great. So everybody who was developing a different approach to encouraging the body to attack the spike protein, the mechanism of action was shown. So that was great. The viral vector was easy to produce, relatively so. You could codify it in the way you wanted it, and you can tune and tailor it, which might be important for variants as we go forward. The mRNA is delivering that message directly into the cytoplasm of the cells without having to go into the nucleus. And and then it self-destructs. That's a bit Mission Impossible, actually, if you think about it in that way. Your mission, should you choose to accept it, once the RNA has delivered the payload, it goes away and the message is done. So those two broad technologies at the forefront of the COVID response have been knockout. The ones that are coming down the line are more traditional based vaccines. They're proteinaceous in nature. They may be VLP in nature, the Novavax, the Valneva type ones. But as you may have heard yesterday on the health briefing by Secretary of State, actually combinations of those two things might actually be required for long-term booster stuff.
Yep.
But let's go back. So I just wanted to say on that. So in this particular case, the proof of mechanism's there. Now, however, massive challenges in manufacture. As ever, when you're faced with a crisis, you do what you know, and quite rightly, the pharma companies went after volume because we've got to do 7 billion people. And they went for technologies which they could scale readily and easily and quickly. And they didn't give much consideration to the delivery aspects of it, and relying on what still is a very high bar to beat, which is a needle and syringe. Yeah, it's easy. People know it. It's familiar. But yes, indeed, some of the challenges there, look at the mRNA. We have either ultra cold chain, -80, very cold chain, -40, compared to the AstraZeneca one, which is 2 to 8, fridge temperature. At Enesi, we've done adenovirus-based vaccines, which are thermally stable at 40 degrees.
Right.
We're basically now doing RNA vaccines at a similar temperature level. So we think that some of our research programs will be able to impart benefit on the future of the COVID vaccines of today, and particularly the next generation ones that come. Proteinaceous things we do are really falling off a log for us, to be perfectly honest. And we get that thermal stability combination with needle-free. Of course, because we don't have to reconstitute either, we don't have a diluent. You may have seen also, Razin, in there was a big hoo-ha about 6 doses in a 5-dose vial. Well, that's because syringes, you can't get everything out, and you lose a little bit. And that might not sound too much of a problem, but on 100 million doses, it means you're only treating 80 million people. That's a big problem. So really what you want to do, and I'll go back to how I described it, are we unique? We're doing thermally stable, unit solid dose guaranteed, so we don't have those wasted doses. But let's summarize at the end. That was it's been a long answer to a question. I'm so sorry about that. Is each and every vaccine has a different robustness. Some are a little more fluffy, some are hard as nails, and accordingly you have to vary your processing to do that. But once you get to your bulk, that's only still 1/15 of the story or so. You've got to package it, you've got to deploy it, you've got to do the logistics. And then it's not so much the last mile, it's the last 5 millimeters, the last 1/4 of an inch that you have to really, really worry about. And I think that's companies such as ours in the novel vaccination delivery space have a really strong part to play.
Right. And is it just vaccines that you can use ImplaVax for, or are there other types of drugs that you can deliver?
Yeah, that's a really good question. That's a little bit Marty McFly, actually, a bit Back to the Future, to be honest.
Well, I love all these references. I'm going back through the ages of all the movies. In fact, I was thinking of The Incredible Journey when you're talking about putting a tablet in Billy there as well. So that's even an older reference than Back to the Future.
Well, actually, The Incredible Journey, that is real technology now. That's the pill camera, essentially, right? That happens. You can do that now. That's some military technology come through. But so that's a really good question, because I think it does highlight actually the limitations to our technology, and every technology has a limit. We've got to be honest on that. So the main limitation we have is we have a fixed payload. So our implants have a maximum mass of 4-ish milligrams, and when contained within that payload, we can only really have a ceiling of 30 to 40% of active material.
Right.
And if we go above that, then we start to lose the structural integrity and strength we need to guarantee implantation. So what does that mean? Well, any chemical entity which maybe have a dosing regimen of less than a milligram or less than a milligram and a half. Yeah, sure, fine, no problem at all. So highly potent materials work really well.
Right.
And actually, to be frank with you, are damn sight easier to work than vaccines, because we don't necessarily have to work in aseptic conditions. You can think about terminal sterilization, and a chemical entity doesn't generally lose potency through processing, because you maintain its purity throughout. So yeah, there have a few things being done. And again, if listeners are on, we're always willing to consider potent materials, albeit the focus of the company when we started was to take the technology into we thought an area of gross unmet need, but actually could have a material impact going forward. The chemical entity stuff also facilitates more for administration at home, be that osteoporosis or growth hormone or something like that. So it's a different concept in terms of value proposition for the recipient of the product. But yeah, for sure. So we can't do everything, and we do have some limitations.
Right. Well, you mentioned value proposition, so that leads on nicely to my next question, which is obviously how cost effective is it using your method compared to traditional methods?
Gosh, that's a very good question as well. And the needle and syringe obviously is a very high bar to beat, because it's mass produced, things are established. And then interestingly, in that type of logistics scenario, people tend to focus on syringe costs or needle costs, or actually the procurement cost of the vaccine itself.
Right.
They are missing a trick there. I mean, even if you look at the National Audit Office for COVID-19, it's only about 1/3 of the cost to date has been on the purchase of vaccines, and 2/3 are actually on the deployment of vaccines. So you have to compare and contrast on a delivered dose basis, which I think is important when you take everything into consideration. So we were always very conscious of that, actually, Raz. And when we started off the business 4 years ago, I did go and spend time in Geneva, actually, with the World Health Organization. Sat down with their pre-qualification team, because not only did we believe the platform should and could and would be available to the developed world, we also wanted a single platform for worldwide distribution. So there are limitations about what you can do in packing weight and volume and stuff. So we learned a lot there. And then we also worked with an organization called PATH out in Seattle, who are very close to the Gates Foundation, whom we also work with. And they have something called a vaccine technology impact assessment model. You can find this on the internet. It's a really good tool. Basically, they ran the runes over us, and then we did a comparison. They believe that analysis shows that actually, despite the sophistication of the ImplaVax system, so the solid dose format, the cassette, the reusable actuator, it came out that we should actually be the least cost per delivered dose on a fair comparison basis anywhere in the world. And for that, we got very enthused about it, because, when you talk to people, or you talk to government, you talk to NGO, talk to military, all those people you work with, they want to know cost of goods. You want to know how much it's going to cost. And actually the modeling going forward is very encouraging. Difficult bit is, of course, you've got to invest in the capital to get you to that point to start with. So on the device aspects of it, we've got a great partnership with one of the world's leading medical plastics company. Our production capability is coming up in the UK. We're pleased about that. We've invested quite heavy in that, at least to get us to this clinical stage. And then, of course, the chemical engineer that's cut me through and stick a rock, like me, is it's all about scale out technology. Scaling up is difficult. Scaling out is easy. Spaghetti making is easy. Just make a longer piece of spaghetti, right? So all of those things come through. And so I think we have a very positive contribution on cost of goods, but also when you start to think about the other things, when we don't have the cold chain logistics, it's very simple to use, a lower skilled person per unit. It doesn't matter if the electricity goes off in my village in Oxfordshire or the village in Uganda, the material isn't going to go off. You're not going to have to destroy it. You've not got wasted doses. And I think that's many of the themes. Folk like us in the delivery space, they're all trying to impart all of those benefits into their approaches to delivery in order to get people to move from the past. Because I said at the top, if you've always done what you've done, if you always do what you've done, you'll always get what you've got. But times We're back to Bob Dylan now. The times are changing, aren't
It's
they?
funny you
And I think they are.
I thought you were about to say that. She said times. I thought anybody's going to say changing.
Yeah, sure.
All right, well that's brilliant. So if people want to know more information, where can they get more info?
As ever, the website, please. www.enesipharma.com. We're also on Twitter. We're also on LinkedIn. I have a LinkedIn profile as well. We try and keep our news up to date. And if I may, we've got 3 press releases coming out next week in our progress on various programs. So encourage your listeners to have a look at those. But it's been a delight spending some time with you today. I'm sure we could talk about many other things other than vaccines, but it's a privilege to have been with you today, and thank you for your time.
Yeah, no, well, it's lovely. And actually, before we go, we will talk about one other thing, because people may be wondering why I'm wearing my orange Dolphins jersey today. One, because it's my corporate colours, but 2, it's NFL draft day today. And we were talking before we started this call about you thinking about Dan Marino and the 80s
Yeah.
NFL and Channel 4. So obviously I'm wearing this because it's draft day today, and that is the day that NFL franchises have the chance to restart their team and hopefully build for the future. One thing we also talked about was the ESL, or the European Super League, which you're a Liverpool fan, which I'll forgive you for, and I'm a Man United fan. And we also talked about how football, though, particularly in Europe and the UK and South America, is not a franchise model, but is very much a city societal model, I think, a community model. And so that's why the ESL failed, because you can't Americanize soccer, as America would like to say, in Europe and the UK, because the fans just would not go for it, because it just kills all sense of competition, and it doesn't really work. You can't. There's only one club, as far as I'm aware, which has ever moved, which is Wimbledon AFC, which is ironically my local team, which has moved to Milton Keynes. But then the fans started another Wimbledon AFC to make up for the fact they went. So, you know, I think the clubs are really ingrained in it, and so I hope the ESL never comes to fruition in that format anyway, where it's a closed shop and it's a franchise model, because I don't think it'll work. I'm sure you would agree with me on that.
I would do. I can certainly forgive you for wearing orange in a high definition Zoom call, but I do remember the days of Dan Marino back in the 80s when Channel 4 brought it on. Of course, what I can't forgive you for being a Man United fan. But as I said to you as well, if there was a ballot, you'd probably, being located in South London, you would win the closest to Old Trafford fan today, right?
Yeah, I'm sure it would be. Yeah. Well, I'm sure that's not true, actually. I'm sure lots of polls have proved that's completely untrue. There's more Man United fans in Manchester than City fans. But anyway, and the only other thing we were going to say was actually that I sincerely hope that Liverpool don't make the Champions League. But I do think, due to City and Chelsea doing so well in the Champions League, and they're in the FA Cup final, that actually the odds of Liverpool going through default is actually very high, because there's a chance that you'll get in because everybody else has won trophies and got in that way. So you can actually move up the table even if you're 7th.
Well, we'll see. I mean, I actually have been looking at that. I know West Ham lost last night, which was very helpful for us. I found myself cheering on both Chelsea and Man City over the last 2 days, and they're both looking quite tidy. And if that's a final, of course, it's going to come down the list a little bit. But there is a cutoff, I think, for maximum number of teams from one country in the Champions League. But, you know, whether we get in by default or otherwise, as I would say, you know, unlike Manchester United, but certainly in the case of the Dolphins, class is permanent and form is temporary. Yeah.
Yeah. Well, we'll have to agree or disagree on who's got the class, though. But anyway,
Right.
but on that note, it's lovely talking to you, David. Genuinely, it's been really interesting learning about the technology. It's a really fascinating subject. I think the whole area of drug delivery, particularly for the future. I mean, I know we touched on Star Trek and, you know, and various other things, but actually just shows how things have moved on since those visions in the 60s and the 70s of how you can deliver medicine and treatments to patients. And I think it's a very exciting future. And I think COVID has accelerated a lot of that development and technology because of the speed the vaccines have come to market. And I think it's exciting times in that particular space. So thank you very much for sharing that information. And I look forward to seeing the developments of Enesi in the future.
Wonderful. Thank you very much for your time. Let's keep in touch.
Well, definitely. And viewers, as always, I hope you found that useful. Check out the links I'll put above the video. If you've got any questions for David, I'm sure he'll be delighted to answer them, either by leaving a comment below the video or messaging him directly on LinkedIn. And as always, until next time, if you're an NFL fan, enjoy the NFL draft. I'm sure there'll be fans who are not happy about who got picked, but just remember, you've got no control over it. Just enjoy what you can and enjoy the rest of the football season as well if you're following that as well. And until next time, stay well and stay safe. Bye-bye.